Hi, everyone. My name is Meera Bhat. I am a rising senior studying biomedical engineering at Johns Hopkins University. And this summer, I was fortunate enough to be in Dr. Rong Fan's lab at Yale University as a part of the NIH SenNet CUSP internship program. So today, I'll be talking about exploring cellular senescence biomarkers in human lymph node as well as humanized liver. So to begin, a little bit about what is senescence. So senescence is the cellular non-proliferative state, and it's implicated in the development of various different age-related diseases. There are many causes of senescence, including one being SASP, a senescence-associated secretory phenotype. These are factors that are released. There's also cell cycle arrest, in which various phases of cell cycle development cause the cell to be arrested. There's DNA damage, as well as other factors such as DNA methylation. But today we're going to be talking a little bit about some senescence-associated biomarkers that I was able to explore over this past summer that are traditionally associated with senescence. And these are P16, P21, gamma H2AX, as well as HMGB1. So some of the pathways of senescence are reasons why senescence occurs within the cell. This can be due to cell cycle arrest, the activation of the DNA damage response, changes within the nucleus of the cell itself, upregulation of anti-apoptosis within the cells, inflammation that occurs, as well as various other forms. But we'll be focusing on three main ones today. We'll be focusing on the DNA damage response, any oncological or cancer-related stressors that occur, as well as those SASP factors that I was talking about, which lead to inflammation within the cell. The way we did this over the summer was in the SenNet Tissue Mapping Center at Yale University. We had two main projects that we discussed, one of which was exploring cellular senescence biomarkers within human lymph nodes. The other of which was exploring cellular senescence biomarkers across a variety of different species. These range from mouse liver to human liver and humanized liver. So to begin, we are going to look at an 86-year-old formalin-fixed, paraffin-embedded, otherwise known as an FFPE human lymph node sample. This is that we analyzed for the presence of cellular senescence. So to begin by looking at the H&E staining, this is the basic lymph node as it was given to us. To note some prime anatomical features of it, we start over here on the left with the primary follicle. This primary follicle is the preliminary state of germinal centers within the secondary lymph tissue. It is essential for adaptive immune responses within the lymph node itself, and it mainly consists of naive B cells as well as follicular dendritic cells. We also go over here to the germinal cells. These germinal centers and germinal cells are the site of activation and proliferation of B cells and B lymphocytes within the lymph nodes itself. And finally, we have the sinus histiocytes over here, and these are specialized in antigen presentation as well as phagocytosis of additional debris and pathogens. Whenever these cells accumulate in the lymph nodes, it's typically due to some form of a reactive immune response to a foreign material or debris that's coming in from outside of the lymph node. So after this H&E staining was done, we did cell typing, otherwise known as single cell integration. This single cell integration is where we're able to see different clusters of types of cells. So over here, we have some memory B cells as well as activated B cells in the orange, and these are all clustered together along the follicles. We were also able to see some CD8 cytotoxic T cells, which are here and outlined in purple, as well as CD4 plus helper T cells in green outside the perimeter of the lymph nodes themselves. By doing this, we were then able to further analyze this human lymph node using a technique known as co-detection by indexing, otherwise known as CODEX. CODEX utilizes various types of DNA conjugated antibodies, as well as the cyclic addition and removal of different fluorescently labeled DNA probes. It's been used to simultaneously visualize up to 60 different biomarkers, and it allowed for us to have a deeper view into some of the single-cell spatial relationships within this 86-year-old human lymph node tissue that we analyzed. We began by initially staining that FFPE tissue with the antibody panel of those 60 different biomarkers before we performed a multi-cycle reaction where we imaged 3 different antibodies and added a nuclear stain to this cycle. After this was done, we stripped the chemicals from them, we hybridized them, and we imaged the actual panel itself with all of the antibodies in place. After this reaction was completed, we computationally processed all of these different images, and from there, we performed data analysis. As a result of the codex, we were able to see a wide variety of different images. We started here with an initial region of interest, ROI1, and we were able to analyze CD20 and CD21, which are known B lymphocyte markers, as well as CD3E, which is a known T cell receptor lymphocyte, and as well as that being a protein. From there, we're able to see a strong expression of CD21, so some of those B cells on the perimeter of the follicle, as well as seeing some T cells on the outside of it. And this gives us a great visualization of these different biomarkers. But of course, to talk about senescence, we wanted to look a little bit more into those senescent biomarkers that I had mentioned beforehand. So that being P16, P21, HMGB1, and gamma H2AX. So when we look at these follicles at the exact same spots, we're able to see an extreme expression of different factors such as the gamma H2AX, as well as the P16 being strongly highlighted within these follicles. Even when you zoom out and look at the lymph nodes, you see the strong presence of different senescent biomarkers such as P21 and gamma H2X on the perimeter of the lymph node, showing that this 86-year-old aged tissue definitely has the expression of these senescent biomarkers. When we did this with a different region of interest, we were able to see even stronger results. Over here, we're seeing the B cells being even strongly expressed within the follicles on the other side of the human lymph node, and we're able to see how the T cells are on the outside of the follicle as well. When we look at the senescent biomarkers, we see an even stronger expression, where factors such as HMGB1 even have a stronger role to play. And overall, it just speaks to the importance of seeing how these senescent biomarkers are strongly present within this aged tissue of 86 years old. After this initial imaging was done, we were able to then do some spatial analysis. So we began by doing UMAP clustering. UMAP clustering is especially important because it helps us to cluster the cells into different groups. So we have the cluster of 1 being the activated T cells, we have 0 being B cells, 3 being B cells and dendritic cells, and we have more from there as well. So this clustering is able to clearly identify which cells are present within the lymph node. From there, we were able to analyze and do a spatial clustering as well. This allows us to take that ROI2 image and be able to see where exactly each cluster of cells is present. So we see on the outer perimeter, we have these green ones that are highlighted here, and these are known to be B cells and dendritic cells. As we progress through the follicle, we see some more B cells here, but it quickly transitions to activated T cells. So we see how the composition of the lymph node is going from a simple codex image into some of the more spatial analysis afterwards. After that, we were able to do a heat map, and this really outlines all of those different biomarkers that we were able to stain for and be able to see how we can identify those clusters. So cluster 0 was remarkable because it mentioned CD20 as well as CD21, which are known to be these kind of immune cells as well as B cells. Cluster 1 as well had activated T cells, and that was marked by CD4. Cluster 2 had B cells and dendritic cells, which were marked by CD44 and CD45. Cluster 3 had macrophages and dendritic cells as well, and that's due to CD68. And then cluster 4, because of CD31 and 163, had the presence of T lymphocytes as well as endothelial cells. So we were clearly able to differentiate between some of the genes and the biomarkers we stained for, as well as the clustering within the cells. And afterwards, when we really looked into the senescence biomarkers, we were able to develop an image feature plot, which shows the strong levels of expression of the senescent biomarkers P21, HMGB1, and gamma H2AX. We were able to see a strong expression within the follicle from each senescent biomarker to another, and it really clearly shows that senescence plays a role within the aging of this 86-year-old lymph node tissue. So from there, we wanted to look a little bit more into the applications of cellular senescence within a humanized liver model. So this was a different project that we explored throughout the summer, and we were able to start out by seeing a humanized liver. This liver consists of both human parenchymal cells, which consists of hepatocytes, as well as non-parenchymal cells, which are non-hepatocytes within the liver, but they support the cells of the liver to aid in their functions as well. From there, we were able to use the technique deterministic barcoding in tissue, otherwise known as DBiT-seq. This is a proprietary technique developed by the lab I was in, Dr. Fan's lab, and it's a microfluidic technique which is used in spatial transcriptomics to deliver barcodes to the surface of a tissue slide. This allows for spatial omics sequencing with 10 millimeter pixel size, and it creates detailed maps of gene expression within the tissue samples. It starts by overlaying a tissue slide with antibody-derived DNA tags. After this is done, the process of barcoding occurs, which I'll explain further, in which barcodes are flown in different directions orthogonally on top of the tissue slide. This allows the barcodes to attach to various different RNAs at their specific location, which is important for developing a transcriptomal map later on. After that is done, we microscope it and image the cell and the tissues within. And then from there, we're able to go and build a DNA library, which is important for sequencing later on. This sequencing allows us to have the tissue image at the initial scale, the mRNA transcriptome that we're building from the barcoding process, as well as the protein panel overall. So a little bit more about this barcoding process. We have glass slides that are in both a perpendicular and parallel direction, and we flow the barcodes from one direction to another. The way this works is we attach a vacuum to pull the barcode solutions, and they're able to directly attach to the RNAs at their specific location. So first we go in one direction, and then orthogonally we go in the other direction, and that's able to directly pinpoint the location of those RNAs. After this was done, we were able to build a spatial transcriptomic map. And this is really important because we're able to see both the parenchymal cells as well as the non-parenchymal cells and how they cluster within the immunosuppressed mouse humanized liver that we're able to see. So we see clusters ranging from mouse hepatocytes within this map. We see mouse macrophages, human macrophages, and even mouse neutrophils. So we're able to see that there's a strong clustering that we can overlay on top of the direct tissue slide imaging. From there, we were able to really look into the P16 senescent biomarker. And we see that on the left here is the mouse spatial heat map. And we see that while the mouse spatial heat map and the human spatial heat map have a very similar expression level, we see that they're at different spatial locations. And so we're able to see that these senescent biomarkers play a role on both the mouse as well as the human cells within this humanized liver model. From there, we wanted to go and see whether we can pinpoint these senescence pathways within the humanized liver. So I mentioned a variety of different senescent pathways that have a role to play within the cell, but we really wanted to focus on three main ones. The first of which being DNA damage, when DNA happens in the cell itself. Number two is when oncogene activation happens, and this leads to the proliferation of cancer cells. And number three is when the SASP, or senescence-associated secretory phenotype factors, are activated within the cell itself, causing senescence. So by averaging genes from a gene set known as SenMayo, we were able to compile the top hundreds genes present within this humanized liver model. From there, we were able to see within each of the pathways a very strong level of senescence being present on that image that we had taken beforehand. Within the transcriptome, we were able to see that by averaging the genes together, there is a very strong presence of senescence within these humanized liver cells in this immunosuppressed mouse. So we were able to see clearly the role of senescence as we go from one pathway to another. So overall, we had a couple conclusions to make. Number one being that the cellular senescence biomarkers, P16, P21, HMGB1, and gamma H2AX were heavily present in 86-year-old human lymph nodes. We were also able to see that human parenchymal cells and non-parenchymal cells that are generated within an immunosuppressed mouse have the presence of senescent biomarkers such as P16. And we also noted an overall increase within the senescent genes across the various different pathways, whether it's DNA damage response, oncological stressors, as well as the SASP factors that I was mentioning within the humanized liver model. Finally, I wanted to give my acknowledgements for this past summer. I had an amazing experience, and this was truly due to the NIH SenNet CUSP program for giving me the opportunity to learn about senescence this past summer. I also wanted to thank Yale University, as well as Dr. Rong Fan of the Fan Lab, for offering me the chance to go in his lab and to learn the different technologies that they have on a much deeper scale. And I finally want to give my biggest thank you to my mentor, Negin Farzad, for being there for me every day and for guiding me through these different wet lab and computational processes that I learned. So I'm very grateful to this program and for the opportunity that I had. Thank you so much.